Hello students, I hope all of you are doing great. Welcome to your own channel, Vedantu NEET English. This is Basavaraj Sir, your biology master students. Students, before we start today's amazing class of biomolecules, I want all of you to tell me in the chat right now if I'm properly audible and properly visible to all of you students. Quickly in the chat right now. Now, if I'm properly audible and visible, add any emoji, students. Add any emoji in the chat to let me know if I'm properly audible and visible to all of you students. Okay. So students, first of all, good evening all of you students, good evening. Right. Now, this is the chapter which is the foundation chapter, and this is a chapter which if you properly understand, it is very easy. Okay. Even if you feel this chapter is not easy, I will make it easy for you students. Now, who can watch this lecture right now? Any student. A student doesn't have to know any concept of this chapter because I will be teaching from the basics to basics. What is lipids, what is protein, everything from basics. So if you are excited and if you are ready for today's amazing class, quickly mark your attendance by smashing that like button. And students, we will start. Students, we will start. All of you. Good evening, students. Good evening, all of you. Good evening, good evening, good evening, good evening, good evening. Okay. Now, students. Now, first and foremost, students, in my class, I always ask questions. So if you are in my class, it is your duty, it is your responsibility to answer to my questions. Okay. Now the first question is very simple. The first question is very simple. I want all of you to name 3 different organisms in the chat. Go. Students, 3 different organisms in the chat. All of you. In my class, 3 different organisms. Let's say a fungi, a human, a tree, or bacteria, protista, amoeba, that is a protista itself, right? So all of these organisms, it could be a single celled prokaryotic organism, a bacteria, cyanobacteria, mycoplasma, all the way to a multicellular tree, multicellular human, multicellular animal. Are we all made up of same chemicals, students? Do you think all of us are made up of same chemicals? Me, you, all of us around, are we made up of a same basic chemical, students? Same basic chemical. The answer is yes. The answer is actually yes. That is, we are made up of, students, all the living organisms, we are made up of something called as biomolecules, students. We are made up of biomolecules. Now you will ask me, sir, what is the meaning of biomolecules, students? Biomolecules are basically the biological, also called as biological molecules. These are carbon containing molecules. Carbon containing molecules involved in metabolic pathway or metabolic process. So all of these, students, all of these are different types of molecules. They are carbon based. They are present in our body in different types of metabolic pathways. Now students, I need one volunteer from the chat. Students, I need one volunteer. Who wants to volunteer, students? Who wants to be the first volunteer of today's class? Anyone? Who wants to volunteer, students? Anyone? Students, who wants to volunteer? All of you. Who wants to volunteer? Okay, let's take Hema. Let's take Hema Varshini. Let's take Hema now. Hema. Now, students, let's take Hema. Now, on this earth right now, students, on this earth right now, let's say this is our earth here. Okay, we'll make it blue color. Right, we'll try to make it blue color because earth is a blue planet. Okay. Now, students, here Hema's grandfather has a property, students. Hema's grandfather has a property here. Okay. So grandfather has a property here, students. We will take a rent from there. Now we will take a piece of earth crust. From this particular region here, we will take a piece of earth crust here. Earth crust will take a piece from it. Okay. From her grandfather property. Okay. Now students, when we analyze this earth's crust, we notice that this particular earth crust has some basic elements. It has some basic elements, students. I know, Sharmita. There are some basic elements here. Okay. This earth crust has something called as hydrogen. it has oxygen, it has carbon. Okay, okay, I agree. Now students, I need one more volunteer. Students, I need one more volunteer. Students, I need one more volunteer. Let's take Sharmita only. Okay. Now we'll take one. We will make a small piece on Sharmita's ear. We'll take a small piece from her ear. Okay. And we know when we analyze the earpiece of your Sharmita, we notice that in the human body also, in the living organisms also, the same. In the case of a cell, let's take a cell right now, students. In the case of a cell also, right? In the case of a cell, the same elements, that is hydrogen, oxygen, carbon, the same thing is present. So here also we have the same thing. Here also we have same thing. On the Earth's crust, we have the hydrogen, oxygen, carbon, and on the cell also we have hydrogen, oxygen, carbon. Then what is the difference? Then students, what is the difference? Here also we have the same thing, all the same thing. Then what is the difference then? Is there any difference? Yes, there is. The difference is in the relative abundance. Students, the difference is in relative abundance. That is quantity. That is, this hydrogen, oxygen, carbon, it is more in the case of the living organisms. More in the case of living organisms. Students, is that clear to all of you? So the same elements which are present on the earth crust and here the relative abundance, the quantity, students, the quantity is very, very high here. Very, very high. Very, very high here. Okay, students, is that clear to all of you? The first concept is clear, students. Nandini, clear all of you? All of you. The first concept is clear to all of you? Yes, it is clear to all of you. Now, students, now whatever molecules are present inside this cell, they are also called as cell pool. They are also called as what? Cell pool. So, I will call this, whatever present on the inside is called as cellular pool or cell pool or cellular pool. Now, inside the cell pool, we have certain inorganic molecules, inorganic elements, right? And we have organic elements inside the cell pool, students. Now, inorganic elements could be your minerals, it could be vitamins, it could be your water also here, water also, students. So all of that are present here in inorganic, minerals and everything. Now, organic part, students, right? Organic part, let me just change this to light pink. Okay. In this organic part, we have mainly two types of molecules. One is called as macromolecule, one is called as micromolecule. So macromolecule and micromolecule, this is what we'll be studying in today's class, students. In today's class, we are trying to understand what are the different types of macromolecules and micromolecules present inside a cell. Present inside a cell, students. Okay, present inside a cell. Okay, students, is that clear to all of you, students? Students, is that clear to all of you? Sir, can we get NEET in this year? Yes, you can get NEET in this year, obviously. Students, is that clear to all of you? So in this chapter, that is biomolecules chapter, we will be studying different types of macromolecules and micromolecules, which are your organic elements present inside a cell. We'll be studying that, students. We'll be studying that in today's chapter. Okay. Now, what are these macro and micromolecules, students? Can you even give me examples? Any few examples here and there? Anyone? Anyone, students? Any few examples? What are these molecules? Okay, I'll give you a clue, students. I'll give you a clue. I'll give you a small clue, students. They are present in this burger. All of these molecules are present in this burger right now. Burger. Students, how many feel of burger? How many feel of this burger? See here. Here you can see the carbohydrates are present on top. We have the nucleic acid present in this this thing. Basically, it is plant-based. Plants have cells. Cells have nucleic acid, genetic material. Right? This is your, let's say this is a chicken burger. This is a chicken burger. In this chicken burger, we can have protein is present here. Then there is cheese here. Cheese is basically lipid. So technically speaking, all the different types of biomolecules are present in burger only. Okay. That is, we have lipids, we have proteins, we have carbohydrates, we have nucleic acids, Everything is present right here, students. Everything is present right here. And here is one more, students. We have carbohydrates, lipids, proteins, nucleic acid, students. Students, which one is your favorite? Students, which one is your favorite, students? All of you, which one is your favorite, students? Which one is your favorite? And among all of them, see, my favorite is protein, because protein, students, building block, no, building block. So I love protein. All of you might like, if you like cheese, we can go for lipid also. Protein is there, but yeah, my favorite is protein, students. My favorite is protein. It looks also very goofy, no? Looks also very goofy. Okay, students. Okay. Now, students, we will do some experiment right now, students. All of you, I want all of you to wear a lab coat right now. All of you, imagine, students, all of you all of you wear a lab coat right now, and let's do a small experiment, students. Okay. Now, in this experiment, we will be requiring a organic substance. So, students, it is your call now. Should we take a leaf or should we take a rat liver, students? What should we take? All of you tell me in the chat, students. Students, what should we take? Should we take a rat? Should we do RIP rat or should we take leaf? It's your call, students. It's your call. Okay. Now we will do a experimental proof that is called as chemical analysis. Okay, chemical analysis, students. Now for this chemical analysis, you can take a liver piece if you want. Papa RIP for rat has to die for this. We can take a liver, rat liver, or if you do not want to kill a rat, okay, we can take a leaf also. We can take a leaf. Leaf is very powerful, no? So we can take a leaf also, we can take a leaf. Either of two works. Okay. Now either of two works. Okay. Now students, what you need to do is you need to take this leaf here and you're supposed to add it into a mortar and pestle. Students, do all of you know what is mortar and pestle? Basically chutney making process. Okay. To this mortar and pestle, we add the leaf or the liver, any one we add. Now after adding it inside, let's say since I'm a botanist, I will take a leaf. Okay, I'll take a leaf here, students. Now to this, which students, tell me, do you make chutney in your house? All of you students. Do you make chutney in the house? So do you make chutney in the house without water? No. We always add water when you try to make chutney. Coconut chutney, mint chutney, any chutney. Yes or no? You add water. Water is a solvent there. Yes or no? Solvent is always added. So here also we need a solvent that is called as trichloroacetic acid. Okay, the solvent here, let's add a solvent, students. Let's add a solvent inside. Solvent is added, students. Solvent is added. That is trichloroacetic acid. Acetic acid. Okay. Trichloroacetic acid has been added, students. Okay. Now, once you add the trichloroacetic acid, what do? You nicely grind it. Okay, if you want, you can use mixie, but we will not use mixie now. We will use this only, mortar and pestle. Now, this is how the mortar and pestle looks. This is how the mortar and pestle looks like. This is how it looks like. Okay, so this is how it looks like, right? This is how it looks like. So we add here, we add the leaf, we add the trichloroacetic acid, and we make the chutney nicely, students. Chutney. Okay. Now, once we make the chutney, students, what is the scientific name of chutney? Anyone? What is the scientific name of chutney, students? The scientific name of chutney here is called as what? It is called as slurry. Okay, students, respect to give the name. We get the slurry from here. Okay, we get the slurry here. Now this right here is the slurry. Green color chutney we got here. Nice chutney, nice. All your skills in the kitchen has been shown here. Okay, so we get something called as slurry. Slurry is okay, students. Now you need to take this slurry. Now we have to do one more process, students. Now we have to do one more process here. Okay. Now we will do one simple process right now. Let's take one container here. Here you have a container, students. Let's say beaker here. In this beaker, on top of this beaker, students, on top of this beaker, here I will add a muslin cloth or a cheesecloth, basically for filtration. Basically for filtration, we add a cloth on top. Okay, we add a cloth. We can add a cheesecloth or a filter cloth. Let's say filter cloth. we can add here. Now on top of this we add our slurry here. Slurry, students. We add our slurry on top here. Now students, all of you tell me, is it possible that some amount of slurry will drip down like this? Tip tip. Yes or no? Some amount of slurry will drip down and it will make a layer here. Yes or no? Yes. Now students, I want all of you to understand one simple thing right now, students. Simple thing right now. Okay. Now, yes, song. All of you sing the song. Dosa, idli, chutney, chutney. Yeah. Okay. Sing the song. Now students here. Now please, this is very careful. Now whatever is present at the bottom here and whatever is remaining part on the top, they are called, they have two different names. Whatever is present on the top and whatever is present on the bottom, there are two separate names for it. Okay. Please listen to this very, very carefully right now. Okay. Now, whatever is present on the top, it is called as retentate. Retentate. Whatever is present on the bottom here, it is after filtration, students. After filtration, it is called as filtrate. It is called as what? Filtrate. So we have retentate and we have the filtrate, students. Filtrate here. Now, students, this particular top, on the top, which is present on the top, right, it's called retentate. Technically speaking, it is present on the top because it was insoluble. Students, it was insoluble in acid, right? Which acid? Trichloroacetic acid. So whatever is present on the top was technically insoluble in the acid. That is why it's present on the top. So can I call the retentate as, acid insoluble fraction? Acid insoluble fraction, students. I can call that acid insoluble fraction. Now, whatever present here which is coming in the downstairs here, dripping, I will call it acid soluble. It's called as acid soluble, students. It is acid soluble. Okay. Now, students, any doubt till here? No doubt here. Now, students, I want all of you to understand whatever is present on the top, their molecular weight is generally more than 1000 Daltons. Now, what is present on the bottom here? Since they are able to come through this filter paper, it means that their size is very less. Okay. Now here, molecular weight is less than 1000 Daltons, around 800 generally. Now, students, now, if their molecular weight is more on the top, do I call them macromolecule or micromolecule, students? All of you, do I call it macromolecule or micromolecule? All of you, since they are present on the top, 1000 atoms more, I will call them macromolecules. What is present on the bottom, I will call them micromolecules. So here we have macro, bottom we have micromolecules. Micromolecules at the bottom. Okay, students. Now, if these are macromolecules, it means they are very long chain. Yes, no, students. They are very, very long chain. If they have to be very long chain, they have to be generally polymers. Yes or no? They have to be generally polymers. And these are technically the monomers. Students, how many of you understood? This is a simple experiment. This is a simple experiment to tell us the retentate and filtrate, the difference. Acid insoluble fraction, molecular weight is more than 1000 Daltons here. Macromolecules, polymers. Acid soluble fraction, molecular weight is less than 1000 Daltons. Micromolecules, and they're technically the monomers. Long chain of monomers here. Long chain of monomers. Okay, they are called as filtrate, students. How many of you understood? Raise your hands. Students, how many of you understood? Raise your hands. Quickly raise your hands. This is a simple experiment to differentiate between macromolecules as well as the micromolecules, students. Micromolecules. Which part you didn't understand? Ask me, students. All of you. Macromolecules are the molecules. Macro means what? Macro means large. Micro means small. So these molecules generally are large molecules, students. Large molecules. These molecules are very tiny molecules. They are very tiny molecules. Is that clear to all of you, students? Micromolecules means small molecules, large molecules here, because their size is more than 1000 Daltons, less than 1000 Daltons. Very simple. Acid insoluble fraction, because they are present on top. It means they are completely insoluble. They are insoluble here. They are soluble. Okay. Acid soluble fraction. Okay. The retentate and filtrate, students. Retentate, filtrate. Okay. Done, students. Done. Students, you can ask me all silly doubts. I always encourage silly doubts, students. Here. Now, students, here is a table here which all of you can see. Micromolecules, examples, simple sugars. When I say simple sugar, I mean monosaccharides, disaccharides, maximum. Okay. Amino acids, nucleotides. So all of these are examples of micromolecules. Now, students, if you look at the macromolecule examples, here we have complex carbohydrates, which includes what? Which includes the oligosaccharides, polysaccharides. Students, do not worry if you do not know what is oligo, everything I'll teach you, don't worry. Okay, yes, students. Now, here we have your proteins. Proteins are very long chains. Here we have nucleic acids, DNA, RNA, everything is here. Yes or no? But students, here there is one KD here, one Kalla here, okay, who is hiding. You know who's that, students? The hiding person here, the scammer here, students, is the lipids. Lipid is the scammer here. Do you know why? Because technically speaking, lipids molecular weight is around 800 Daltons. Lipid is what? Lipid is, students, lipids are, right? Lipids is basically molecular weight is less than 1000 Daltons. 1000 Daltons. Now then why are we kept in macromolecules? Then why are they macromolecules, students? Why are they macromolecules? If their size is less than 1000 Daltons, why are they present? Why do we have lipids in? Why do we have lipids retentate? Why are lipids present here? Anyone? Why is lipid present, students, here? Why is lipid present there? Students, how many of you played this game called as chain game when you were kids? Students, why do we add acid? Do we add acid for lysing? There is something called as see, the cell has cell wall. Yes or no? The plant cell has cell wall. It has plasma membrane. So we add acid to break the layers and everything. Okay. Tell me, students, how many of you played the game called chain game? Students, how many of you played a game called as chain game? Now, same game is played by your lipid also, students. Lipids also. Chain game. So, what lipid, students, will lipid do? Lipid will form a vesicle. Students, even though each individual lipid is very tiny, but what will it do? All the students, all of these lipids will hold hands. They're like, do not leave me at all. I am your life, you are my life. Okay, so all of them will hold hands. So when they try to hold hands, they form vesicles, and these vesicles do not pass through. Okay, so these vesicles will not pass through here. So all of these vesicles are present on the top itself. All lipid vesicles are present on top. That is why even though lipids are technically less than 1000 Daltons, but then also they are present here in retentate. Is that clear to all of you? Power of unity, exactly. Power of unity. All of you, show some unity in the class. Okay, students, it's a very amazing game. Chain you should play. You go, you do chain cut, and you run. Okay, so play that game. So that is the power of unity shown by lipids, students. Lipids. Okay, now, students, here's a table for all of you students. Here you can see the comparison of Earth crust and human body. Students, can you see here very clearly? Hydrogen is less in Earth crust. Human body more hydrogen. Carbon is less. Carbon is more. Oxygen is less. Oxygen is more. Nitrogen is less. Nitrogen is more. So in the starting, I told you, right? In the starting, I told you here that These molecules are present high inside a living organism and less in the case of earth crust, and it is also given in your NCERT. Here's a table, student. Here's a table for all of you. Okay, here is a table for all of you, student. Here's a table for all of you. Okay, now students, now can we do one more experiment? Students, shall we do one more experiment right now? Shall we? All of you students, quickly smash the like button, all of you. Students, ecology will come after this. Ecology, you want ecology, I'll do ecology, don't worry. Ecology is coming up. Students, shall we do one more experiment right now? This experiment is very easy, very tiny experiment. Okay, it is called as ash test. Okay, it is called as ash test. Now, in this ash test, we basically try to find the inorganic molecules. See, because of this test right now, because of this test, we got the organic molecules. Now, same test can be done. One more test can be done to isolate the inorganic molecules also. Okay. Now, for this, we need a fresh leaf. Okay. We need a fresh leaf right now. Okay, fresh leaf is here. Now you're supposed to take this fresh leaf and completely evaporate it. That is minus H2O. Okay, students. Tell me, will the fresh leaf become dry leaf now? Yes or no? It will become dry leaf right now. Fresh leaf became dry leaf right now. Very simple logic. Dry leaf right now. Here we had something called as wet weight because water was present here, and this can be called as dry weight because water is absent right now. Wet weight and dry weight, students. Wet and dry weight. Now, students, you're supposed to take this nice dry leaf and you grind it in a mixie. Vroom, vroom, full mixie you add the grind. Students, once you grind, you get what? You get the powder. Now what do? You burn the fire. We burn the powder. Completely burn this powder, students. Now once you completely burn the powder, what do we get? We get the ash. That is, here there is removal of any type of water. Everything is gone here. Any type of CO2 is also released to some extent. Evaporation has happened, oxidation has happened, everything has happened, and we end up getting the ash. Students. We end up getting the ash here, right? Now this ash can be used. This ash can be used to do the chemical analysis. Now, once you do the chemical analysis with the help of other instruments, we get the list of all the inorganic molecules present inside, students. Okay. So when you do the ash, like a Pokemon ash, okay. So when you take the ash, you get all of the different types of your inorganic components also. Water is obviously not found here because water was evaporated. But apart from water, we can find all of these different types of your inorganic elements. So these two different experiments shows you that a living cell, a living organism has organic molecules. Same organism also has inorganic molecules, students. Is that concept clear to all of you, students? Is that concept clear to all of you? Students, upcoming videos have tomorrow. I'm taking the part 2, students. Tomorrow only I'm taking the part 2. Okay, students, is this clear to all of you? Students, is this clear to all of you? Everyone in the chat, is this clear to all of you? The organic and inorganic molecule. Here is a list of all the inorganic molecule present in the living system. It is given your NCERT itself. It is given your NCERT. Students. So if let's just summarize what we learned till now. We learned that components of life has organic components and inorganic components. Organic components with the help of the first experiment, right? First experiment, we got the biomolecules and biomacro and micromolecules, and with the help of ash test, we got the inorganic molecules also. So with that, the first part of the chapter is over. Students, with that, the first part of the chapter is over, students. Now, students, I have a new information for all of you, right? Now I'll be confirming, right? I will be showing you NCERT side by side. So we know that we have covered every single line of NCERT, students. Okay, so we learned that we started off with the biomolecules in general. We learned that all organisms are made up of different same elements, right? Same elements. If you perform the such analysis on plant tissue, animal tissue, or the microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several other elements. Now, same experiment, students, same experiment can be done on the Earth's crust also. Earth's crust, yes or no? We get the same element. What is the difference, students? The difference is very simple. That is, a closer examination reveals that relative abundance of carbon, hydrogen is more. Okay, session is not over. We have session is still 1 more hour is there. 1 more hour the session is there. Okay, a closer examination reveals that relative abundance of carbon and hydrogen with respect to other element is higher where? In living organism. So a living organism has higher elements. Same thing, higher quantity, abundance. Okay, then analysis, students, analysis. This is your experiment. We took a vegetable or a piece of liver, trichloroacetic acid, mortar and pestle, cheesecloth, we ran it down. One we got filtrate, other or more technically acid soluble fraction. All of you, we got the filtrate or acid soluble fraction here. Same thing, yes or no? See, fraction. Scientists of have found thousands of organic molecules in the acid soluble fraction. Right. Now, in higher classes, you will learn about how this analysis can be done. Right. Everything is given here, students. Everything is given here. Okay. So there is a definition for biomolecules. All the board students, here is the biomolecules definition. All the carbon compounds that we get from living tissue is called as biomolecules, students. Here is a definition for all of you. Right. Here is a definition for all of you. Definition. Okay. Students. Here is a definition for all of you. Biomolecules definition. If you're class 12th. Okay. Then students. Then we have what? Then we have the ash test. Ash test. That is burning, incineration, more dangerous students. Once we do the ash test, we get to know hydrogen, different elements are present, students. Okay, different elements are present. Okay. So with that, we have finished till here right now. Now we will start with the amino acid, students. Now students, we'll start with the amino acid. Any doubt till now, students? Any doubt till now? We learned about the basics, we learned about the experiment, and we learned about the different elements present in a living organism. Organic and inorganic, students. Organic and inorganic. Now, now students, let's study amino acid, students. Let's study the amino acid right now. Okay, let's study the amino acids right now. Amino acids. Okay. Now students, when I say amino acid, what is coming to your mind right now? Amino acids, when I say amino acids, what is coming to your mind right now, students? In your mind right now. Amino acids technically are, students, very small or very large. Students, I told you, amino acids, right? Amino acids are present in the filtrate. They are present in acid soluble fraction. They are called as micromolecules. Yes or no? They are called as micromolecules. Their size is less than 1000 Daltons. Size is less than 1000 Daltons. Those are your amino acids, students. Now, amino acid is technically a monomer. Now, if I take one amino acid, one more amino acid, one more amino acid, and one more amino acid, and if I keep joining them, students, when I keep joining them, what do we get? What do we get, students? Multiple amino acids, when we keep on joining them, we end up getting what? We end up getting the protein. We end up getting the protein, and protein is a macromolecule. Protein is a macromolecule. So these are micromolecules. Here, micromolecules. Yes, sir, students. We get the micromolecules. That is your amino acid, students. Now, there are so many amino acids. There are so many proteins in the world. There are so many different types of protein in the world. Right? You can see, students. You can see. Millions are there. Millions of proteins are there. Millions and millions of proteins are present in the world. To make this millions of proteins, how many amino acids do you think there are? Anyone? Anyone, students? There are so many proteins, so obviously we need to have many amino acids, right? Obviously we need to have many amino acids, but not. Students, okay. Let me change the question. Students, how many words do we have in English language? Students, students, how many words are present in English language? Tell me, how many words are present in English language? There are so many words. Words keep on adding all the time. For this entire words, millions of words, how many alphabets do we have? Students, how many alphabets in English language? All of you. Very simple question. There are millions of words. To make this millions of words, how many alphabets do we have in English language? All of you. How many alphabets? 26. Yes or no, students? Or 27 now? Who's student 27? Huh? Go back to UKG. Students, how many alphabets? 26 alphabets. Now, just like these 26 alphabets can make up millions of words in our body or in nature, we have around 20 amino acids. Only 20 amino acids. These 20 amino acids will make up all the protein in the world. Crazy, no, students? Crazy, no? So, 26 alphabets, millions of words. 20 amino acids, millions of proteins. That is the power of amino acid. That is the power of amino acid, students. Amino acid. Now, question arises. Where do we get this amino acids from? Students, where do you think we obtain the amino acids from? Okay. Some amount of amino acids is produced in our body. Some amount of amino acid is produced in our body itself. Our body is producing. Now, what about the rest? Students, where do we get it from? Anyone? Where do we get the protein from? Chicken biryani. Students, chicken biryani, mutton biryani. Yes or no? Paneer, students. Paneer we can take. We have dal. We have all of these are called as what? Protein sources. These are called as what, students? Protein sources. So all of these can be used to derive the proteins, students. Now, as I told you, some proteins is made, some amino acids is made by the body. That is called, students, that is called as non-essential amino acids, students. Because non-essential amino acids are the amino acids which are produced by the body. They are produced by the body, students. Produced by the body is only making it, students. The body is only making it. Okay. Now, students, what is essential amino acids? Essential amino acids are the amino acids which our body does not make. Okay. These are the amino acids. These are the amino acids which our body does not produce. If our body is not producing it, then how do we get, students? These amino acids, essential amino acids, need to be taken through diet. That is the food which you eat. Through diet and food, we take up all the essential amino acids. So non-essential is already produced in our body. Essential needs to be taken from outside, students. Now, students, I'll tell you one big scam right now. All of you, one big scam. Students, ready for the big scam? Students, do you know there are many people who go to gym? Yes or no? There are many people who go to gym. When these people go to gym, students, they always take something called protein powder. Have you noticed? They have big boxes, even I have in the house. Okay. So all of this protein powder, right? Imagine, students, this protein powder which they are taking from outside. Imagine there are multiple companies which are always scamming them, because general public do not know. Because general people without who are science background, they do not know. These companies sometimes will always add all the non-essential amino acids and give you in the box. Then I go take, be happy. You are like, oh, I am taking protein, I am getting amino acids in my body, you're very happy. But what is the reality? Reality is that you are technically only taking in what? You are only eating what? Non-essential amino acids. Students, is there any use? Students, is there any use? Is there any use if you take only non-essential amino acids? No. So next time if you know any of your friend, if you know any of your friend who goes to gym, or your parents or your brother, sister, anyone, tell them to check the box. Tell them to check the box if they have added the essential amino acids, because these are important for our body. These are important for our body, students. Okay, these are important for our body. See students, the meaning of good diet is nothing but you take a diet where you have good protein source in your food. That is all is dieting. Dieting doesn't mean fasting. Fasting and dieting are two different words. Fasting is you do not eat food. Dieting is that you control the food items which you eat. Difference is there. Okay, so extra information for all of you students, extra information for all of you. Okay, so here are all the list of essential amino acids, non-essential amino acids, students. List is present here. Now you will be like, sir, very difficult to remember. Do you need a trick, students? Do you need a trick? Again, this is not my trick. I stole it from somewhere. Again, this is not my trick. I stole it from somewhere for all of you students. For all of you. Imagine so much of KD Kelsa I'm doing. You know what is KD, students? KD, right? Kala Kelsa I'm doing for all of you. I am going everywhere else, stealing the examples, and I'm coming for all of you. See, be happy in life. Okay. So what is the, students? What is the hack here? The hack here is that essential amino acid is private Tim Hall. Okay, right? It is called as private Tim Hall, right? Private Tim Hall. Here we have phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, lysine, leucine. So all of these are called as your essential amino acids, students. Essential amino acid. Okay. Now this is called as non-essential amino acids. Almost all guys get cut after going to play. Again, students, in examination they generally don't ask this. Students, again, in generally they do not, students, they do not ask this in general. They do not ask. But by chance if they ask, just a little bit have a look at it. Okay, have a look at it, students. Have a look at it. But students, here is a problem. I'll tell you the problem. If you don't practice the trick will play a trick on you in exam. If you do not practice the trick, no, the trick will make you fool in the exam only. How? Understand, students, understand. Okay, let's say here we have A for arginine, here we have A for aspargine or A for aspartic acid. Now imagine you write aspartic acid here instead of arginine. So that can happen, right? That can happen, yes or no? So please remember what you're learning, because in the exam you'll be like, Basavaraj Sir told this, all wrong answer to write and come. Okay, so please be very careful. Please be very careful. Okay, please be very careful in life. Okay, now students, now we'll play a small game, students. Now we'll play a small game. Now we'll play a very, very small game. Okay, now technically amino acid, students, technically speaking, amino acid is substituted methane. It is a substituted methane. Can anyone tell me what is the formula for methane? All the chemistry people in the chat, all of you, Telugu people, all of you chemistry people in the chat, tell me what is methane. All of you tell me what is methane. Tell me what is methane. I'll complain to Diksha ma'am now. Otherwise, will raise my complaint against you. Tell me what is methane formula, students. All of you students, you're not sharing the session, huh? Is it? Students, all of you are not sharing the session. Huh? Yes. Methane is CH4. Yes or no? CH4. Okay. So we have methane here. Carbon, hydrogen, 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 and hydrogen. Okay. Now students, very simple game. Okay. I will give you this hydrogen. You are supposed to give me one functional group. Okay. Now I will give you this hydrogen. I'm substituting it. You are supposed to give me carboxylic acid. Students, what is carboxylic acid? COOH. Okay. Now students, I'll give you this hydrogen again. I'll give you this hydrogen, students, but I need an amino group. Amino is NH2 group. So now I will give you this hydrogen again. You're supposed to give me R, that is functional group. And here we are, students, here we have the, what do we have? The basic structure of amino acid. Here we have a carboxylic acid group, yeah, amino group, and with that, done, students. Difficult or easy, students? Difficult or easy? Easy, no. Now, this R can be anything. This R can be hydrogen. This R can be hydroxymethyl. It can be methyl. It can be ethyl methyl. It can be anything, right? Now, depending upon what this R is gonna be, there are different amino acids. Okay. Now, this is called as a basic framework. Okay, this is called as a basic framework, students. In this basic framework, you just have to replace the R right now. Let me give a few examples. Students. Okay. Now let's say we draw the we have carbon. So this is an alpha carbon, right? It's an alpha carbon because it has functional groups on the sides, right? Alpha carbon, alpha amino acid. Here we have the COOH. Here we have the NH2. Here we have the hydrogen, and here we have the R. Now, students, if this R is going to be hydrogen, let's say, if this R is going to be hydrogen, right, this R is going to be hydrogen, in that case, this amino acid, students, is going to be glycine. It is going to be glycine. Done. Glycine, students, glycine. Students, please remember, this is a glycine right now. Okay. Now, let's say this R right now, students, is going to be a methyl group. If it is a methyl group, in that case, this amino acid is going to be alanine. Okay. Now, let's say this R is going to be CH2OH. Students, what is CH2OH in the chat, all of you? CH2OH is hydroxymethyl. Okay. In that case, this amino acid is going to be serine. So here we have a gas. What do we have here? Gas here. That is glycine, alanine, serine. Glycine, alanine, and serine, students. 3 different things here. Okay. Yeah. What do you want, students? Something and all, sun, moon telling. Students, clear? All of you. Now, students, does anyone know what is chiral compounds? I will not go very detailed, but I'll tell you a simple outline basic. Do you know what is chiral compounds, students? Chiral compounds? All of you. Chiral compounds? All of you. What is chiral compounds? Yes. Do you know what is chiral carbon, students? Chiral carbon? Anyone? So please remember, students, glycine is actually the simplest amino acid here, and it is going to be non-chiral in nature. Why non-chiral, students? It is going to be non-chiral because here also we have hydrogen, here also we have hydrogen. So here 2 functional groups becomes the same. So it becomes non-chiral. Chiral means what, students? Chiral means all 4 are different. All 4 are different. That is called as chiral, students. Carbon 4 different groups. Now, but in the case of glycine, if you add H here, it becomes non-chiral. It becomes 9 chiral, students. Okay, 9 chiral. Okay, it becomes non-chiral. So notes I'll give you today. Okay, it becomes non-chiral in nature, students. It is also optically inactive. But I'll not go, students. I'll not go so detail. I'll not go so detail also. That you learn chemistry mainly. Okay, students. So we have alanine and we have serine, students. All of you remember this, students? All of you remember this? MS I'll send. MS sir I'll send. Okay, students. Any doubt here? Students, I'm going as slow as possible. I don't want to rush this chapter. If I rush anywhere, you will not understand. Very simple. The chapter is very, very simple. I do not know why everyone says this chapter is very difficult, but it is not difficult at all. Very, very basic, easy chapter this is. Okay. Students, I will share today only. I share today only. I will share DPPs. Today I will share DPPs. Students, just remind me someone in Telegram. Okay, students. Now this diagram you don't need to know. This diagrams this formula you do not need to know. So biology, no diagram I keep saying. Okay, this formula you do not have to know, but just remember methionine and cysteine, both of them are sulfur containing amino acids. Students, have you heard anywhere methionine anywhere? Methionine somewhere in your codon? Somewhere, students, methionine somewhere you have heard? Anywhere you have heard, students? Okay, very simple rule, students, very simple rule. If this video gets 150 likes before I end, tomorrow we'll finish chapter. Today we'll finish chapter, students. Okay. Okay. Students, if I get 150 likes on this video and all of you comment, tomorrow only I'm taking the class. Tomorrow morning, 11 o'clock, we'll finish. Start codon. Exactly, students. Start codon. Methionine. Sulfur containing amino acids, students. So if this video gets good likes, right, and or if you comment, then only I'll give you tomorrow. Tomorrow I'll take 11 a.m. class. We'll finish the chapter tomorrow only. Okay. Now, students, now I want all of you to understand. Students, amino acids can also be in different chain form also. They can be aromatic or they can be aliphatic also, students. Yes, right. Do you know what is aliphatic, students? Aliphatic are straight chain. Aromatic are round. Okay, aromatic are round like benzene ring. All of you know benzene row. So here are three different aromatic amino acids, students. Here are three different aromatic amino acids. We have phenylalanine, we have tryptophan, and we have tyrosine, students. Students, do you remember there was a disease called as PKO disease, right? PKO disease, phenylketonuria disease. In phenylketonuria disease, did we remember phenylalanine there? Did we talk about phenylalanine? Phenylalanine gets converted to tyrosine there. Yes or no, students? Yes or no? Phenylalanine gets converted to tyrosine in PKO disease. In the PKU disease, yesterday's class, PKU disease, phenylalanine becomes something else. Okay. Yes or no? Yes. PKU disease. Students, tryptophan anywhere, students? Tryptophan is actually involved in auxin preparation. In auxin, you will learn. Friends, you want to do plant physiology with me, right? Plant physiology, all of you want, right? In plant physiology, I'll teach you the auxin preparation. There we use tryptophan actually, tryptophan. Then we have tyrosine. Tyrosine, all of you know, the same disease, PKO disease only. Okay, PKO disease only. Okay. All of you, Mendelian disorder. Very good, students, very good. Okay. Students, it is also called as your emergency hormone. All of you should remember. Students, you don't have to remember the structure, remember the examples, that's all. Aromatic amino acids. Okay. Now, students, there are three different types of amino acids. One is called as acidic, one is called as basic, one is called as your neutral amino acid. Now, students, all of you tell me in the chat right now, what makes a amino acid acidic? Is it the carboxylic acid part or is it the amino part, students? Anyone? Students, I don't have Snapchat only. I don't even use, students, I hardly use Instagram also. You rarely see me adding anything on Instagram. I don't add anything on Instagram also. I don't use only. I don't like social media only, first of all. I do not like the social media only. I stay away from social media. Okay. Last time I added a photo was 1 year back, I feel. Okay. Look at the irony. I don't like social media, but I'm on YouTube. Right. Now, students, the acidic part is COOH. Yes or no? The acidic part is carboxylic acid part here. So, in the acidic amino acid, students, what do you see extra here? Students, in acidic amino acid, do you see a extra carboxylic acid here? So, here are the two examples. Here, aspartic acid and glutamic acid, students. Two different types, students. Two different types of your acidic amino acids. This you have to remember. Aspartic acid, glutamic acid. Names you have to remember. Structure, don't need to remember. Structure is for you to understand, students. Structure is for you to understand. Okay. Now, students. Now, if I say something is basic amino acid will have extra what? All of you use your brain cells. All of you use your brain cells, students. Basic amino acid will have extra what? Basic amino acid will have extra amino group. Yes or no? So in basic amino acid we have lysine, arginine, histidine. So here we can see it has a extra amino group here. Extra amino group. Students, do all of you remember, students? Do all of you remember this lysine and arginine? Lysine and arginine are present in your histone protein. Do you remember, students, anywhere? In molecular basis you might have studied. In molecular basis you might have studied, students, molecular basis. Somewhere you have studied, right? Lysine and arginine are present where? Lysine and arginine are present somewhere, right? Okay. Yes or no? Somewhere you have studied. You will remember. Okay. I'm just trying to connect chapters now. I'm just trying to connect the chapters. So we have lysine, arginine, histidine, 3 basic amino acids, students. 3 basic amino acids. Here is the example of glycine, students. Glycine is a neutral amino acid. Valine is a neutral amino acid, students. As a matter of fact, I do have extra slide for it. As a matter of fact, out of the 20 amino acids, 2 are acidic plus 3 are basic. So students, tell me all in the chat, if 2 are acidic, 3 are basic, remaining how much are left, students? Students, remaining how much are left? All of you. Tell me, students, tell me quickly. 2 are acidic, aspartic acid, glutamic acid, histidine, lysine, arginine are basic. Remaining all amino acids, 15 amino acids, students. Remaining all 15 amino acids are neutral in nature, neutral in nature. Done? Okay, done, students. Now, I will teach you a difficult concept, right, students? Right now, I'll teach you a difficult concept. Ready, students? Ready? Okay. I want all of you to pay attention, students. I want all of you to pay attention. Very, 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 very attention. Full attention now. Okay. Students, these amino acids are like you only. I always say this. Amino acids are like you only. When no one is in the house. You'll be like, oh, I have Tattva book. I will keep the book somewhere else. I will take my phone and I will like, oh, Instagram, nice. But, when no one is around, you're chilling, around eating something, random things, chilling. But as soon as dad or mom comes to the house, what do? You will like, you will throw the phone, you'll put one nama, you're like, yes, Papa, yes, Pitashri, you're like, okay. You'll start to act, you're reading now. Oh, you'll start acting. So depending upon, students, depending upon the situation, do you act differently, students? Yes or no? Depending upon the situation, you act very differently. No one is in the house, you're like chill mode, chill today, full chill. Today I will, Basavaraj Sir class is there, I will not attend today. Basavaraj Sir class I'll watch recorded. But as soon as dad or mom comes, you're like, gone. Book, you not even take one book, you will take 10 books. 10 books, you'll keep one big, you'll make a mountain of books as if you're studying from 10 days. Okay. So just like how you behave differently, students, how you behave differently in different environment, amino acid also behave differently in different environment. Now, what do I mean by that, students? What do I, students, understand? Yes, what do I mean by that? Okay. Now let's say, students, let's say one second, let me just delete this. Students, what do I mean by that? Let's take one amino acid right now. Let's take one amino acid. Let's take one amino acid right now. Let's take this is amino acid. Okay. Now this is a normal amino acid. This is a normal amino acid, normal amino acid. Now if I take this amino acid and I keep it in acidic medium, what will happen, students? In acidic medium, do we have H plus everywhere? In acidic medium, do we have H plus everywhere? Yes or no? Do we have H plus everywhere? Yes, we do. Yes, we do, students. Now this H plus is now taken by NH2. So in acidic medium, the amino acid will become basic. It will become basic. Stupid it is. Okay, it becomes NH3. It becomes basic in nature, in acidic medium. In acidic medium. Okay, let's take one more amino acid. Let's take one more amino acid, students. Okay. Now I will keep it in basic medium. This stupid in basic medium, what will it do? In basic medium, it will lose the hydrogen to the environment. So in basic medium, students, in basic medium, it will become acidic in nature. Stupid amino acid. Okay, it will become basic in nature, students. So depending upon right, depending upon the environment, they keep on changing, students. They keep on changing. Okay, right, depending upon the situation, they keep on changing. In basic medium, they become acidic. In acidic medium, they become basic. These are your amino acids, like you, students. These are amino acids just like all of you. They keep on changing, students. But what is the but, students? What is the but? The but here is very simple, students. But here is very simple. At a particular pH, right, something special happens. At a particular pH called as isoelectric pH. At an isoelectric pH, something special happens. What happens, students? Let's say here we have a carbon. Here we have the carboxylic acid. Here we have the NH2. Here we have the functional group. Here we have the hydrogen. At a particular pH called as isoelectric pH. At isoelectric pH, what will happen, students? This particular hydrogen is transferred here. Now that is called as what, students? That is called as intramolecular proton transfer. At a particular pH, there is something called as intramolecular proton transfer. Now, because of this, what happens? Because of this, what happens is that minus here, NH3 plus will become NH3 plus, and here we have hydrogen. So this right now, student, this right now here, which has partial positive charge, students. This has something called as partial positive charge and it has partial negative charge. At the same time, this molecule here at isoelectric pH, because of intramolecular proton transfer, this one right now is called as what? It is called as Zwitterion. So this is called as, students, this right now is called as Zwitterion, or it can be called as amphoteric ion. Amphoteric ion, students. It is called as Zwitterion or amphoteric ion. See, can you see, students, here? All of you here. This is called as Zwitterion right now. Zwitterion, right? Zwitterion right now, which has a partial positive and negative charge, students. Zwitterion formation happens. So what is Zwitterion? Zwitterion is nothing but a molecule with one functional group having positive charge and other functional group having negative charge, and the net charge is going to be 0 here at isoelectric pH. Done. That is called as Zwitterion, students. Zwitterion is formed at a isoelectric pH like this. At isoelectric pH, it is for isoelectric pH. What happens at isoelectric pH? This hydrogen is transferred here, right? Intramolecular proton transfer happens, and because of this transfer, what is happening? There is a Zwitterion formation like this. There is a Zwitterion formation. It is also called as amphoteric ion at isoelectric pH, students. How many of you understood? Raise your hands. Students, how many of you understood? Raise your hands quickly. If you understood, solve this question quickly. This is a NEET PYQ. Solve. All of you. Solve the question quickly. All of you smash the like button first of all. Quickly, smash the like button. Done? Basic amino acid is lysine. Solve. This is a 2024 PYQ. The following can be found as Zwitterion: fatty acid, monosaccharide, amino acid, nucleic acid. Which one do you think will form the Zwitterion? Hello Emperor, long time no see. That's amino acid. That is your amino acid, students. Amino acid. Now, students, how many of you seen this? Not the chakli and the fries, the oil stains behind it, students. How many of you seen this? Students, how many of you seen this? Anyone? Students, how many of you seen this? If you have seen this, what do you think it is, students? What is this thing? What is this thing in the background? What is this thing in the background? Anyone? What is that thing in the background? What is that? What is that, students? What is that? That is oil. Yes or no? That is oil. Oil. Students, this oil can also be called as what? Oil can also be called as lipid. It can also be called as, technically speaking, this oil, ghee, everything is part of lipid, students. Lipid. Now, as a kid, students, have you ever tried to mix oil plus water? Students, have you, yeah, French fry or cholesterol, I know. Have you ever tried to mix oil plus water as a kid? Anytime? Have you tried to mix oil plus water? Okay. If you are trying to mix oil plus water, do you think it mixes? It does mix actually. Initially, it may float. Initially, it may float on the top, but later on, if you keep on agitating it, if you keep on shaking it, the oil will mix with the water, and that is called as emulsion. That is called as emulsion. Okay, that is called as emulsion, students. So lipids weigh less than 800 daltons, but still, students, are present in the acid insoluble pool, students. Okay, why? Because they form vesicles, students. They form the vesicle like this. They form vesicles like this. They form vesicles. Okay, so students, lipids, all of you, present in retentate filtrate, their size is less than 1000 daltons, but they are present where? They are present in the retentate, that is acid insoluble fraction on the top, students. Okay, now students, but these structures which are lipids, they are not forming polymers. They are not forming polymer, remember. They are forming vesicles. They are forming vesicles, but they are not forming polymers. Please, students, please remember that. There is no polymer formation here. There is no polymer formation here. Unlike your nucleic acids, proteins and everything, they do not form polymers. Okay, they are not polymer, students. Okay. Now, students, lipids can be divided of two types. This lipids can be of two types. One is called as fatty acid, other one is called as glycerol. Other one is called as glycerol here. So we have fatty acid and glycerol, students. Fatty acid and glycerol, students. Okay. Now, students, how many of you know what is the difference? Students, how many of you know the difference? Difference between fatty acid as well as your glycerol? Anyone who can tell me? Difference, you know? Students, do you know the difference between fatty acid and between the fatty acid as well as the Glycerol. If you do not know, raise your hands. Simple as that. So simple as that. If you do not know, raise your hands, because I will teach you very slowly, properly, diagrammatically. I'll show you what is the difference between a fatty acid and glycerol, students. If you do not know, raise your hands. If you do not know, raise your hands quickly in the chat, students. Because right now, students, we'll be starting off with the lipids. Okay, lipid students. Okay, so lipid students, broadly speaking, students, lipids generally are divided into three categories. Broadly speaking, we have simple lipids, compound lipids, and derived lipids. Now, these are the simple lipids which are divided into your fatty acids and glycerol. That is the simple lipid only. Compound lipid, derived lipid is not given in your syllabus, but I'll give you few words. I'll tell you a few words here. Do not worry. I'll tell you a few words. Okay. So students, shall we start with fatty acid now? What is a fatty acid? It has to be fat, no? Yes or no? Now students, understand what is the meaning of fatty acid right now. Fatty acid. I will draw the structure. You will understand. Very, very simple it is. Okay. Fatty acid, students. Fatty acid will have a R group and it will have a students, anyone? What is this group called as? Anyone, students? A particular, always remember, a fatty acid always has a functional group here. Along with the functional group, it will always have a carboxylic acid group. So it has a R group here. It can have a R, the functional group, and this R can be methyl, that is CH3, or it can be ethyl, that is your C2H5, or it can be any number of carbons, 1 to 19, any number of carbons it can be. 1 to 19, any carbon can be this long chain of 19 carbons, 16 carbon, it can be there. When you add along with this, we add a carboxylic acid, we get the fatty acid, students. It is called as fatty because of the functional group, acid because of the carboxylic acid, students. That is why it's called as fatty acid. Okay. Yes or no, students? All of you understood? Yes or no? All of you understood what is fatty acid? All of you? All of you understood? Yes or no, students? This is called as fatty acid. We have the R group and we have the carboxylic acid. That's all it's called as. That's all is fatty. That's all is fatty acid, students. But students, if you say classification of fatty acid, there are two types mainly. One is called as your saturated. One is called as unsaturated here. Saturated and unsaturated fatty acid, students. Difference is very clear. Difference is very, very clear. Understand? Saturated fatty, students. Saturated fatty acids will always have only single bond. Long chain, no students, long chain. The long chain always have single bond. While your unsaturated will have double or even triple bond can be seen here. Right? Triple bonds can be seen here in the case of unsaturated, students. Now, while your saturated, students, saturated, remember, their melting point is very high. Melting point is very high and they are solid at room temperature. Solid room temperature. Okay. Now, compared to unsaturated, their melting point is kind of low. They be very fast, students, and they are liquid at room temperature. Liquid at room temperature. Now, I'll give you an example. It makes sense to all of you. I'll give you an example. It will make sense to all of students. How many of you love ghee butter, students? How many of you love ghee and butter? Raise your hands. How many of you love ghee and butter? Ghee, butter, all of you. Nice chapati you'll add everywhere. Students, if you have ghee, butter in your house, right? Tell me, students, this ghee and butter in your house, do you think it is solid right now or liquid right now? Ghee and butter, students. Is the ghee and butter right now in your house solid or liquid? All of you. Ghee and butter is generally solid, right? It is solid, always generally solid. Okay, so here saturated fatty acid, students, is your butter and ghee. Butter as well as ghee is the example. While your cooking oil, students, cooking oil which you use in the house, no, cooking oil, the sunflower oil which you have, that is liquid, no? The sunflower which you add is liquid always. Sunflower oil is generally liquid in room temperature. That is your cooking oil here. One more example they always give is gingerly oil. Students, I have a history. Always, I don't know why. This sounds very funny, you know. Gingerly. Who keeps gingerly, students? Gingerly oil. What even is that word? It's that word every single time I teach this chapter. I always wait for this word to come. Gingerly oil. Right. Right. This is called as your gingerly oil right now. Gingerly oil. Right. Gingerly oil. Cooking oil. That is gingerly oil, student. Gingerly oil. Okay. Now there are other examples also. Palmitic acid, stearic acid, and everything. Palmitic acid, stearic acid, everything is there. Okay. Now, students, now gingerly oil. What stupid name? Who comes up with that name, students? Okay. Ghee and mustard oil. This is your unsaturated, saturated, unsaturated. Okay, students. Okay, students, I think I didn't teach you that part. Okay. I'll try it here only, students. Now here, students, under saturated, right? Under saturated, we have two other oils which you have to remember, right? One is called as palmitic acid. One is called as stearic acid. Palmitic acid is 16 carbon, remember, students. It is a 16 carbon compound. 16 carbon compound. Palmitic acid, students. 16 carbon. Then we have the stearic acid. This is 18 carbon. Now under unsaturated, students, we have the arachidonic acid. This is 20 carbon, students. This is 20 carbon. Okay. So there are different examples in exam, students. In example, they will ask you directly. Okay. In examination, they will ask you directly the number of carbons in palmitic acid, number of carbons in stearic acid, number of acid in arachidonic acid. How many carbons are there? Okay, they can ask, students. They can ask you directly like that. Okay, students. Now, let me ask you something, students. Do you want to know why olive oil is very good for health? And that's why it's very costly also. Students, why olive oil is so costly also? Do you want to know? Anyone? You might have heard, right? You might have heard olive oil is very good for very body. Why, students? Why? Because this olive oil, students, this olive oil has something called, students, it has something called as PUFA. It has something called as PUFA. Okay, and this PUFA, that is polyunsaturated fatty acid, it is very good for our health. It is very good for our health. It is not purified, nothing like that. Nothing like that, students. It has fatty acids. Okay, the only difference is that it has something called as PUFA, and this PUFA will reduce the bad cholesterol in the body. That's the difference. Okay, so what is MUPA and PUFA? Understand. Here, I give you different acids, right? Different unsaturated fatty acids. All of these are different. Under unsaturated, here other examples are there. Okay, oic acid, oleic acid, linoleic acid, linolenic acid, arachidonic acid. Now, all of these will have what? If they are unsaturated, right? If they are unsaturated fatty acids, if they are unsaturated fatty acids, they will have double bond. So here, oleic acid has only one double bond, while your linoleic and linolenic has 2 and 3 double bonds, where arachidonic acid has 4 double bonds. So this one is MUFA, that is mono means one unsaturated fatty acid. So only one double bond is there, while PUFA has many. PUFA has many, students, many. Okay, so polyunsaturated fatty acid. So it has PUFA in it. So it's a good. Students, with that, fatty acid is also over. Friends, with that, fatty acid is also over. Done. Okay. Done, students. That's why we say it's good for health. Generally, we say it's good for health. MUFA. Right. Right. Understand, students? Understand? PUFA basically means there are more double bonds. See, unsaturated means what? First of all, unsaturated means double bond, triple bond is present. Now, how many double bond is present? In the case of MUFA, only one double bond. MUFA, only double bond. While your PUFA has 2 or 3 double bonds here. More double bonds. Okay. Mufasa movie. Oh, nice movie. Mufasa was nice movie. Students, understood all of you? Understood? Mufa and Pufa. Okay. Now, students, we come to something called as glycerol right now. Glycerol, students. Shall we start? All of you. Students, shall we start? Something. All you're learning today. Students, shall we start glycerol now? Yes. Shall we start? Pakka? Pakka? Okay. Now, students, glycerol. Now, glycerol, students, it is a type of your lipid only. Type of lipid. Here, glycerol is made up of your 3 different carbons here. Let me draw and show you. Let me just draw and show you, students. Okay. Nah, Mupa and Pofa, nah. For me, gingerly oil. Gingerly oil is always. Okay. Gingerly oil is always the epitome of laughter for me. Gingerly oil. Okay, students, understand. Now, what exactly happens in the case of glycerol, students? Glycerol are nothing but type of alcohol. They are basically type of your alcohol which are made up of 3 carbon, 1, 2, 3, 5 hydrogen, three OH. One second, let me just write it in. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 5 only. Oh my god. Hydrogen. 1, 2, 3, 4, 5. Plus 3 OH. Students, this has a IUPAC nomenclature also now. Chemistry. This is the structure of glycerol. It is also called as trihydroxyl propane. It is called a trihydroxyl propane here. This one. It's also called as trihydroxyl propane, students. Okay. Trihydroxyl propane, students. Okay. Trihydroxyl propane. Tri-basic alcohol, no? Trihydroxyl propane it is called as. Okay. That is the other name of glycerol, students. Other name of glycerol. Other name of glycerol, students. It is colorless liquid. All of you know glycerol, right? Glycerine you add on the, when you get ulcers, you add glycerine, same derivation. Same derivative. Glycerine also same derivative there. Glycerine, students. It is colorless, viscous fluid, little bit sweetness is there. All that, students. Now that is called glycerol. Now, what is interesting is this glycerol and fatty acid are part of lipid. Now, how can we join them, students? How can we join them? How can we take glycerol? How can we take fatty acid and join them? Anyone? Common name, okay, students. Chemistry, I don't remember chemistry anyway. Okay. So, how are glycerol and fatty acid attached, students? By esterification. Okay. So, let me just draw and show you. Let me just draw and show you. Okay. Let's add glycerol here. Carbon, carbon, carbon. OH, 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 1, 2, 3, 4, 5. Now this side here is glycerol. Glycerol. Now let's take palmitic acid. We will add a palmitic acid. Students, palmitic acid is how many carbon? Anyone? Students, palmitic acid is how many carbon? All of you. All of you. Palmitic acid is how many carbon? 20, 15, 16, 12, 5, 8, 9. All of you. Palmitic acid is how many carbon? Palmitic acid is 16 carbon, students. 16 carbon. We learned here, right? We learned here. Palmitic acid is 16 carbon. So let's write in structure here. Okay. Now 14, 15, right? And carboxylic acid. This is the R group here. This is the R group here. And this is the carboxylic acid group. Now here we have palmitic acid, students. Now we are supposed, students, to this OH from here, and we will add it to first carbon, let's say, and this OH and this H. Students, what do we get? What do we get? H2O. So can I remove? Students, can I remove water? Okay. Minus H2O. You remove water. So once you remove water, students, what do we get? Once you remove this entire water, we can join these two molecules. Yes or no? So let's say carbon, carbon, carbon. Here we have OH. Here we have OH. And here we have H, 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 and H. Okay. Now students, now hydrogen. Now what do we have here? We have oxygen here. Oxygen will bind to this, but right? Oxygen is here. Oxygen is here, students. Oxygen is here. So this was the only oxygen here left. To this, we have to add this C double bond O. Yes or no? C double bond O. Yes or no, students? Double bond O. Then remaining part, CH2 14 times and CH3. So this is what we get, students. This is what we get at the end of esterification. Right? Now this is called as what? Glycerol plus a fatty acid. Glycerol plus fatty acid. We get something called as what? Glyceride. We end up getting something called as glyceride. Glyceride. Now students, I am adding one fatty acid. I am adding one fatty acid right now at one carbon here, for this carbon here. So it is called as monoglyceride. Now let's say, students, here I add this. Now after adding this, I will add one more here. Then what is called as? Then it is called as diglyceride. Diglyceride. Now let's say, students, let's say I added this here. Let's say I added one more fatty acid here, and let's say I add one more fatty acid here. So all three, oxygen has a fatty acid next to it. In that case, it is called as what? Triglyceride. So monoglyceride, if only one you add. If you add one more here, diglyceride. If you add one more here, it becomes triglyceride. So mono, di, and triglycerides. So monoglyceride, diglyceride, and triglyceride, students. Three different types of your glycerides you get. Glyceride. So your glycerol plus fatty acid is glyceride. So all of you know what is glyceride, students. All of you know what is glyceride right now. All of you know what is triglyceride. Yes or no? It is called as triglyceride. Okay, students. Now students, let me teach you something extra. There is something called as compound lipid. Do all of you remember this? Students, I told you, simple lipid is over. Compound lipid, I'll teach you only one. Understand what is compound lipid. In compound lipid, students, compound lipid is a lipid which will have fatty acids. It will have alcohol. So there is fatty acid, there is alcohol. Along with fatty acid and alcohol, they have one more molecule next to it. That is, additional group is also present here. This additional group can be a phosphorus, protein, sugar. It can be anything. Additional group. So fatty acid plus glycerol plus additional group. That is called as compound lipid. One example which is given in your NCERT is phospholipid. In the case of phospholipid, we take a fatty acid, glycerol, and phosphate next to it. We add phosphate. Okay, we add phosphate next to it. Okay, phosphate. And when we add all three, fatty acid plus glycerol plus phosphate group, we end up getting something called as lecithin molecule. Lecithin molecule. Lecithin. So lecithin is a phospholipid present in the cell membrane. So lecithin is a phospholipid which is present in the cell membrane. So all of you know what is phospholipid? All of you know what is phospholipid? Phospholipid is a lipid which is made up of fatty acid plus glycerol plus other additional group. Here in this phosphate, example is lecithin is an example, students. Lecithin is an example. Lecithin is a example, students. Okay. Now under compound lipids, we learned the phospholipid is done. There is something called as glycolipid also. Let me tell you one small thing, students. Students, what does the meaning of glyco? Glycose, glucose. Glyco means sugar. So in glycolipid, it is not given NCERT. First of all, this is not in NCERT, this is in NCERT. These two are not given NCERTs. Okay. Glycolipid will have fatty acid plus glycerol plus sugar next to it, sugar, carbohydrate next to it. One example, students, one example of your glycolipid is galactocerebroside. Galactocerebroside, students, it is the main lipid found in the myelin in the glycolipid called as cerebroside. And this, student, this myelin, where is myelin present? This myelin is part of your white matter of your brain. So we have lipid in our brain also. Technically speaking, we have lipids in our brain, and that is called as galactocerebroside. Yes or no? Crazy, no, students? That is present in our brain right now. Lipids are present in our brain also. Galactocerebroside. White matter is called as because of the whitish fatty appearance. Okay, so white matter because it has whitish fatty appearance, and this whitish fatty appearance of your white matter, it is because of a lipid that is called as galactocerebroside. Okay. Then, students, here compound lipid is done. I told you main phospholipid is main thing here, right? On neuron lipid I'm talking about. Derived lipid, they have not explained in your NCERT, but they have given you example, student. They have given you example. What is the example? Example of derived lipid is cholesterol. Students, cholesterol, tetracyclic in nature. It is a example of derived lipid. So this is given, student. This is given in our body. This is given. So this is given in NCERT, but it doesn't say from where. So remember, cholesterol is a derived lipid, student. Derived lipid. Okay, students. With that, we are coming towards the end of today's class. I have one more topic for 10 minutes, and I'll be done after this. 10 minutes topic, students. Shall we start? I have a 10 minutes topic, and after that I'm done. Because after this I have one more class also. I have one more class after this. I have to teach Olympiad kids. Right? I have to teach Olympiad also. Right? So last topic. Ready, students? Last topic today. You have a question on the board. You have a question on the board, students. You have a question on the board. Who can tell the answer? Who can tell the answer, students? Who can tell the answer? Yes, students, I also teach in DNA batch, all of these, obviously. Students, today we had a DNA batch class, right? Today morning, what did I teach? In DNA batch today, I taught flower. We finished androecium, aestivation. We finished the entire part in DNA batch today. Students, again, DNA batch is going next level. If anyone from DNA batch is in the chat, in the class right now, telling them how amazing I'm teaching in DNA batch. Right, DNA batch, I'm teaching morphology. I'm writing down the notes for all of you. Right, in DNA batch, I show videos, I show animation, and in DNA batch, I show images. I show a lot of images because biology you can't understand by just simply written part, right? I show a lot of images there. Every single concept I have an image to show in DNA batch, right? So, yes or no? How crazy DNA batch is. Every single diagram I show, and teach them properly DNA batch. Okay, students. Question, tell the answer. Tell the answer, students. What is genetic material? Genetic material is, it can be a DNA, it can be RNA. DNA and RNA are called as what? DNA and RNA are called as nucleic acid. They're called as what? They're called as nucleic acid, students. They're called as what? They're called as nucleic acid, students. Okay. It is called as what? Nucleic acid. Okay. Students, nucleic acid is nothing but nucleic acid, students. Nucleic acid is nothing but what? It is present in the retentate. It is acid insoluble fraction. Yes or no? It is going to be what? It is going to be size is more than 1000 Daltons. And next, all of you. Next, nucleic acid is a polymer. So since nucleic acid is a polymer, it needs to have a monomer, right? It needs to have a monomer. Like how protein, students, like how protein is a polymer. Protein is made up of amino acid, amino acid, amino acid, amino acid. Lot of amino acids. Yes or no? So what is the monomer here? In this case of this biomacromolecule, in the case of this macromolecule, what is the monomer? The monomer is going to be nothing but nucleotide. Nucleotide is going to be the monomer, students. Okay, nucleotide is going to be the monomer in the case of nucleic acid. It is going to be the nucleotide is going to be the monomer, students. It is going to be monomer. Okay, monomer right now, students. Monomer. Okay, so students, what is the nucleotide made up of? This all of you know. So I will go over it because you have learned molecular basis, right, from Kobika ma'am. Someday, students, someday I will teach you molecular basis. Okay, so molecular basis, you learn there what is the nucleoside. You learn what is a nucleotide. You learn what is a nucleoside. What is a nucleotide, students? A nucleotide has three components. This is a nucleotide here. It has a pentose sugar. It has a nitrogenous base and it has a phosphate. So pentose sugar, nitrogenous base and phosphate is present. All three together is called as nucleotide. Now if I remove this, students, if I remove this, what do we get? If I remove this, we get a nucleoside. We get a nucleoside then. Very simple, very simple. This is nucleoside. In nucleoside, we have the nitrogenous base, we have sugar, but no phosphate here, students. There is no phosphate here. That is nucleoside. Okay, students. Now, what about the pentose sugar? Which type of pentose sugar is present? Okay. Now, we have two types of pentose sugar, students. The pentose sugar, there are two types. It is a 5-carbon monosaccharide here. Monosaccharides here. We have the ribose kind and we have deoxyribose kind. This ribose kind is present in the case of RNA and deoxyribose kind is found in DNA. Now, students, if you tell the full form of DNA, what is full DNA? Full form of DNA is what? Deoxyribonucleic acid. Deoxy. The name deoxyribonucleic. Deoxyribo is coming from where? It is coming from the sugar part. So, students, if you see the difference here, if you see the difference between, students, difference between deoxyribose and ribose, difference is very simple. Difference is that in deoxyribose, in the second carbon here, in the second carbon, one oxygen is damar. It is missing here, missing. But in the case of ribose, students, in the case of ribose, here we can find a oxygen. That is the main difference, students. So deoxy, D means what? D means missing. Oxy means oxygen. In this ribose sugar, one oxygen is missing in the case of deoxyribose, and it is found in the case of your DNA. It is found in the case of RNA, students. RNA. That is the pentose sugar difference in RNA and DNA, students. Pentose sugar. Now, what about nitrogen base, students? There are totally 5 nitrogen bases: adenine, guanine, cytosine, thymine, uracil. Students, do you think all of these 5 are present in your DNA? Or one is present in your RNA, one is present in DNA, students? Do you think all, students, do you think all 5 of them are present in DNA? No, students, no. Yes. Okay. It is not present in DNA at all. Not present in DNA, students. So we have something called as purines and this something called as pyrimidines. Now here there is a small trick here. Okay, small trick, students. Small trick is pure as gold. Pure as gold, students. Pure as gold. Okay, pure as gold. That is purines. A for your adenine, G for guanine. So adenine and guanine are purines. They are purines, students. They are purines. Okay, that is purines. 2 of them are purines here. Purines. Now, what is pyrimidines? Pyrimidines are cut the pyramid. Cut the pyramid. Cut is what? C is for cytosine. U is for uracil. T is for thymine. So cut pyramid, we have all the pyrimidines, cytosine, thymine, uracil. Pure as gold, we have the guanine and adenosine, students. Now, students, please remember here one small thing. Okay, please remember that purines are always double ring structure. Purines are always what? Double ring structure. Purines, double ring structure. Purines are double ring structure. While pyrimidines are always single ring structure. Okay, so pyrimidines are always single ring, while your purines are double ring, student, double ring. Okay, and students, please remember again one more time, pure as gold, cut the pyramid, done. Nucleotide will have all three, nucleoside will have only sugar and base, nucleoside will not have phosphate. Okay, students. Now bonding, here students, in the case of one nucleotide to next nucleotide, one nucleotide to next nucleotide, the bonding is going to be phosphodiester bond. That is 3' to 5' here, 3' to 5', we have the phosphodiester bond. This bonding you will learn more in molecular basis, but not here. Just remember, between the two nucleotides, we have the phosphodiester bond. Between the two amino acids, we have peptide bond there, peptide bond. Phosphodiester bond is present. Okay, now students, here, see this, students, see this. So in the case of DNA, we have your thymine, cytosine, guanine, adenine and guanine. And in the case of RNA, students, can you see, in the case of RNA, thymine is replaced by uracil. So in the case, students, in the case of your RNA, thymine is replaced by uracil. Now students, here is something called as Chargaff rule. All of you might be knowing Chargaff rule. That is ATGC. All of you know, students? All of you know ATGC. 2 hydrogen bond among your ANT, 3 hydrogen bond among guanine and cytosine, students. Now students, please remember, this is present in the case of DNA. In the case of RNA, students, AUGC. AUGC in the case of RNA, students, because thymine is replaced by uracil. Thymine is replaced by uracil, students. Uracil. Now, students, now please understand. Adenine, purine, thymine, pyrimidine. Always remember, purine will always form bond with pyrimidine. So purine and pyrimid will always form a bond. Purine will not form a bond. One purine and one pyrimidine, they will form a bond always. GC, right? Right. So that is it, students. With that, here is one more thing which is given in NCERT, students. NCERT is given in NCERT. Carbohydrates, all of you know, which I'll teach you later on. Amino acids, glycine, alanine, serine, glycerol. Students, what is glycerol called as? What is the other name of glycerol, students? Trihydroxypropane. Palmitic acid here, 16 carbon, yes or no? 16 carbon. Can you see? Phospholipid, lecithin. Can you see? This lecithin has a phosphate group here. So it has fatty acid, it has glycerol, and it has your phosphate here, 3 things. Then your triglyceride, it means every single oxygen is one fatty acid here, second fatty acid here, third fatty acid here. Triglyceride. Cholesterol is a derived lipid. Okay. See, purines, adenine, pyrimidine, uracil. Always, students, in nucleoside we always write uridine, adenosine, INE for nucleoside. But for nucleotide, we always write ic acid, adenic acid, right? Adenic acid. So ic acid is told for your nucleotide, nucleotide. Okay, nucleotide, students. Students, last topic is called as metabolites, students. Last topic is called as metabolites. Very, very simple. Here nothing to study. Only one table I'll tell you to study. Okay. Please remember, students, please remember, plant will produce something called as primary metabolite and secondary metabolite. Primary metabolite is required for the plants for all the activities. Primary metabolites are your carbohydrates, lipids, proteins, nucleic acids. Plants require them for direct growth. But secondary metabolites, they do not require directly. These secondary metabolites are released by the plants for stress signal, also along with the stress signal for defense mechanisms. Defense mechanism. Color, pigments, alkaloids, rubber, antibodies, gum, essential oils, scents, spices. So all of these are called as secondary metabolites. Secondary metabolites produced by the defense purpose or stress response. That's what you have to remember. And students, you have to remember this table right here, students. This table right here. If you remember this table, today's class is done. Okay, second implementable students. Pigments means what? They give color. Carotenoids, anthocyanin. Alkaloids, you have morphine and codeine. Morphine, the drug, right? Codeine is in the cough syrup generally. Terpenoids, more terpenes, diterpenes. Essential oil, students. Nice smell you get from plants. Essential oil, lemongrass oil. You put it everywhere and you apply on your skin also. Essential oils. Lemongrass oil. Toxins, arbutin and ricin, students. Ricin is a toxin. If you watched a show called as Breaking Bad, if you have watched a show called Breaking Bad, in a Breaking Bad, he makes this toxin from mustard. From mustard, he makes a toxin called as ricin there. Concalvaline A is a lectin here, which can also help in cell cycle. It helps in cell cycle also. Vinblastin and curcumin longa. Vinblast, students. Vinblastin is a cancer treating property. It has cancer treating property. And this vinblastin is obtained from vinca rosa. Curcumin from curcumin longa, that is turmeric. Then polymeric substance, rubber, gum and cellulose. All of you know, students, all of you know. Okay. So students, with that, we come to the end of today's class, students, end of today's class. Students, again, I'm reminding you again, students, if you are NEET 2027 aspirant, then the best thing for all of you is the pro batch. I've told you always, students, pro batch will take care of you for that exact 2 years. Unlimited doubt solving. You have doubt anytime, anywhere, doubt. Take a photo, upload, answer gets in. Person will call and check on you. That is the pro batch. That is for 2 years. But if you're already in 12th right now or a dropper, then you can always go for a DNA batch. Okay. Students, again, I'm telling you, there I'm teaching already. I've finished 4 chapters, I guess, or 5 chapters in DNA batch already, students. 5 chapters in DNA batch. Okay. Students, do you want tomorrow? Do you want this part 2 tomorrow or not? Do you want tomorrow part 2 or not, students? Part 2 tomorrow morning, 11 a.m. All of you. Do you want the part 2 tomorrow itself, or do you want it after 1 month? Or after next week, basically next week? Tomorrow itself, students? Tomorrow itself you want? Okay. So students, I'll give you a homework question. Right. All of you have to come tomorrow class, 11 a.m. Tomorrow 11 a.m. I have a class. If all of you show up, then I'll take a class tomorrow. Otherwise, I'll not take. But there is a homework question for all of you. Very simple question. I want all of you to answer. Okay, in the comments, not here. In the comments, I want to see everyone's comment. I want to see every single one's comment. Then only I'll take tomorrow's class. Okay. Question is very simple, students. In the comment section, first of all, students, you have to comment. Right. You have to comment the aromatic. No, let's do it. You have to comment acidic amino acid. You have to comment the acidic amino acid in the comment section. Which are the 2 acidic amino acids in the comment section? So while you're typing the answer, you can also type. It'll not take away any money. Type how was the class also. Okay, students. So try typing how was the class also, students. Students, evening I can't take because evening there's one more class. Evening there's one more class in the evening. Okay. 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 So let us know in the comment section what are the 2 acidic amino acids in the comment section, students. Okay. So thank you so much, students. Bye bye. All of you take care and have a great day ahead. Right. All of you take care ahead. I can't take in the evening because Govicam is taking the evening. There's no slot tomorrow evening. Evening slot is not there. Okay. So students, bye bye. All of you take care and have a great day ahead. I hope all of you are doing great. I hope this session was useful to all of you. So please study. Bye bye, students.